So the second part of this lecture on the cell cycle is going to focus solely on M phase or mitosis, which is the division of chromosomes as well as the cytoplasm of one cell to finally produce two new cells. And mitosis or the M phase happens after G1, S, and G2 phases have been completed. And the new cells produced via mitosis or M phase at the end will be identical to the old cell. And so these daughter cells are going to have the same number of chromosomes and they're going to be identical to the mother cell, right? So mitosis generates two identical daughters. And just a brief review on chromosomes and DNA before we talk about mitosis. So chromosomes are one single molecule of DNA in a double helix form. And what chromosomes are basically just tightly packed DNA. It's a way to take the 6 feet of DNA that are in every cell and make it more compact, easier to transport, and easier to divide into two new cells. And so in order to pack DNA into chromosomes, DNA first has to wind around proteins called histones to create this beads on a string structure. And then that beads on a string structure folds and loops together to condense and coil and ultimately forms sort of this X-shaped chromosome that we're all familiar with. So after S phase, when a chromosome has been replicated or doubled, chromosomes have this characteristic shape, which you can see here. So before S phase, you can imagine that there's just one half of the X. And then once that chromosome has been replicated, it forms both halves of the X after S phase. And there are some structures that we're going to discuss that are important in mitosis. And so this whole structure here, this whole X, is one chromosome. It's a set of two sister chromatids, one half of the X and the other, which are two strands of genetically identical DNA or chromosomes, right? So the one on the left is identical to the one on the right because it's been produced during S phase when the DNA was replicated. And the two sister chromatids of a chromosome are held together at the center by a structure called the centromere. Right? So two chromatids making one chromosome held together by the centromere. And during mitosis, those two sister chromatids, or these two identical strands of DNA, are separated, and each one can go into one of the new daughter cells and become the chromosome in these cells. Each species has a different number of chromosomes. Humans have a total of 46, organized into 23 pairs, right? And so we have two copies of chromosomes 1 through 22, as well as an X chromosome. So 23 pairs or 46 chromosomes in total. And because most cells have two copies of each chromosome, or a pair of each chromosome, they're what's known as diploid. And so diploid being di and diploid being two copies of each chromosome. And one of these chromosomes comes from your mom, and the other one comes from dad. So you have a paternal set and a maternal set of chromosomes. And so each pair, for example, so this chromosome 1 here, will have a paternal and maternal chromosome as part of it. All right, and so to get into how these chromosomes are ultimately divided into new cells, we're going to talk about the stages of mitosis. And mitosis is divided into 5 stages: prophase, metaphase, anaphase, telophase, and then cytokinesis. And we're going to go into the details of each of these stages in the rest of the lecture. So to start out in M phase, we begin with the stage called prophase. And in prophase, the DNA has to condense into chromosomes. And so you can see here in this cell, in interphase, the DNA is in blue, and it's really loose and stringy and spread out within the nucleus of the cell. During prophase, that DNA is condensed into the X-shaped chromosomes that we're familiar with. In addition, the envelope around the outside of the nucleus that holds all the DNA in dissolves. And that's to allow the chromosomes to move freely about the cell, which they'll need to do for the later steps in cell division. And additionally, the cell needs to form a structure called the mitotic spindle that's going to be helpful in separating the chromosomes into the two new daughter cells. And what the mitotic spindle is a structure made of cytoskeletal proteins that divides the chromosomes during mitosis. You can see here a cell that has the DNA labeled in blue, or the chromosomes labeled in blue, and the cytoskeletal mitotic spindle labeled in green. And there is an organization of the mitotic spindle at areas called centrosomes. And each mitotic cell has 2 centrosomes, and all of these cytoskeletal filaments, all of these spindle fibers are organized there, and they can be attached to chromosomes for the next step of mitosis. Right? And so you can see the fibers of the mitotic spindle here and the chromosomes in blue. And in this phase of mitosis, the second phase or metaphase, the spindle fibers are attached to the chromosomes. And they do that at that central structure called the centromere. And then what those spindle fibers do is they push and pull these chromosomes, move them around the cell until they're ultimately lined up directly in the center, which is easy to visualize in both of these images here. And when they're lined up in the middle of the cell, this is what's known as the metaphase plate. That's one of the major hallmarks of metaphase. And this is really important. And the transition from metaphase, where all these chromosomes are lined up in the center, to the next stage where the chromosomes are separating into what will become 2 new cells, is one of the most highly regulated parts of the cell cycle. I'd like you to take a second, pause the video, and think about why that might be. Why it might be important to have those chromosomes lined up directly in the middle of the cell before they split. So the reason for that is so that the DNA can be split evenly, because you can imagine that if some chromosomes are on the left of the cell, some are on the right, and then the cell splits into 2, one cell will end up with more DNA and the other cell will end up with not enough DNA. And too much or too little DNA is bad for the cell. And so by lining the chromosomes up perfectly in the middle, we can ensure, or cells can ensure, that they get an equal amount of DNA in each new daughter cell. Right? And so in metaphase, the chromosomes are lined up and connected to the mitotic spindle at the centromere. And then in the next phase, anaphase, the sister chromatids, or the 2 halves of that X-shaped chromosome, are pulled apart. And as they're pulled apart, the mitotic spindle shrinks towards the poles, or the 2 ends of the cell, and basically pulls those sister chromatids along with them, away from the metaphase plate towards the poles. And this basically ensures half of the DNA ends up in this what will be the left cell, and half ends up in what will be this cell on the right. And then after the sister chromatids are separated, the cell will enter into the next phase of mitosis, or telophase. And what telophase is basically just the opposite of prophase. So you'll remember that in prophase, the chromosomes have to condense, the nuclear envelope has to dissolve to free the DNA, and the mitotic spindle has to be made. Well, in telophase, we have to uncondense or decondense these chromosomes. We have to reform the nuclear envelope around them in both of the 2 new cells, and we have to dissolve the mitotic spindle, because now that the DNA has been separated, it's no longer necessary. And so what you'll notice in this cell that's in telophase is that technically this is still just one large connected cell with 2 nuclei. And in order to physically divide this cell, we're going to need to progress to the next phase and the final phase of mitosis, which is cytokinesis. And so at the end of telophase, technically the DNA has been divided, or the chromosomes have been divided, but the cell has not physically divided into two yet. That happens through the division of cytoplasm during cytokinesis, where the one large cell in telophase, which has two nuclei, is ultimately physically divided into two. And so in animal cells, this process is a little bit simpler. An animal cell membrane is squishy, and it can be contracted and then ultimately pinch off to generate two new daughter cells. And you can imagine this as kind of like a big water balloon with a rubber band wrapped around it. And that rubber band, this contractile ring, can pinch down and generate two. Ultimately, those two membranes will fuse together to generate two new cells. But a plant cell has a cell wall around the outside, which is hard, meaning that cell can't pinch off. And so in plant cells, the cell after telophase has to build what's known as a cell plate, which is basically cell membrane and cell wall components that will separate these two new daughter cells at the end of cytokinesis. And so animal cells pinch off, plant cells must build a cell plate to ultimately become cell wall between two new daughter cells. And so you can see a nice outline here of all of the phases of mitosis, as well as nice pictures of the chromosomes and the spindle with these important hallmarks highlighted. And I would suggest using this as a helpful way to study all of the different phases and what important things happen in each of them. And so, for the last part, I just want to ask a few questions to check how well you understand mitosis. So take a second, pause the video, and answer the question regarding which of these stages correctly shows the sequence of stages of mitosis. So the correct answer is C. Prophase comes first, followed by metaphase, anaphase, and then telophase. So chromosomes are condensed, they line up in the middle of the cell, they're divided, and then the chromosomes decondense to form two new cells. This one is a little bit more difficult. So take a second and pause the video and answer this question. If you start with a parent cell that has 48 chromosomes, how many chromosomes is each new daughter cell going to have after mitosis and cytokinesis take place? And so the answer is 48. And this is can be simply answered by knowing that the parent and daughter cells in mitosis are identical. So mitosis generates identical daughter cells from the parent cell. And the way that it does this is by replicating these chromosomes during S phase of the cell cycle. So in order to make sure that a daughter cell has the same amount of chromosomes, S phase is extremely important, or that DNA has to be replicated. Because you can imagine that if a parent cell did not replicate its DNA and then split 48 chromosomes into two new daughters, each of those daughters would only have 24 chromosomes, and that would not be enough DNA to be functional as a cell. So in order to counteract this division of chromosomes every single time a cell divides, the DNA has to be replicated in S phase. And so the DNA chromosomes get replicated in S phase to a total of 96 chromosomes, and then those 96 chromosomes are ultimately split into 48 and 48 in the two identical daughter cells generated through mitosis.